Today's notes are going to be all about biotechnology. So, this word might be unfamiliar to you, but if you just look at what's making up this longer word, we have bio and technology, and I think you're probably familiar with both of these. So, bio, what we're referring to is life, and in particular, we're talking about DNA. Technology, you are definitely familiar with that. So, if we put these words together, or this prefix and this word together, it says biotechnology is applying technology to DNA and proteins, using organisms to create or modify products for a specific use. So, we can analyze DNA or manipulate DNA in order to make food healthier, last longer for us. We can use this technology to solve crimes, to figure out who is guilty of a murder, for example, by matching up DNA found at the crime scene to a suspect to see if they're guilty. We can use biotechnology for cloning purposes. There are many different uses of biotechnology. I think we mentioned all of these. So make medicine, cure diseases, improve crops, make better food, cloning, solve crimes. Lots of different ways that biotechnology can be useful for us. One process that is involved in biotechnology is something called gel electrophoresis. This is a method for separating DNA fragments to create a unique pattern for comparison. So this is used to maybe solve a crime. You can compare DNA from different people and try to match it up with DNA found at a crime scene. So the way this works is that DNA is first cut into fragments, and then DNA is placed into these tiny holes called wells at the top of a gel. Electricity is then applied to the DNA samples, and then the DNA moves down through the gel. Since DNA is slightly negatively charged, it will move down towards the positive end of this electric box that it is in. The DNA is just left to run for a while through this gel, then eventually it's going to separate enough so that you can analyze it and compare different DNA samples. And this is called DNA fingerprinting. So if we look at this example here, it looks like we have some DNA from 3 suspects, and we have DNA that was found in a piece of gum that was left over at a crime scene. According to the DNA fingerprint to the right, which suspect is guilty? So if we try to match up the DNA found in the gum to the 3 suspects, we can maybe figure out who did this crime, whatever that may be. And it looks like suspect B perfectly matches up with the DNA found in the gum at the crime scene. So it's very possible that they're guilty of whatever this crime could be. Restriction enzymes are proteins that are used to cut DNA at specific sequences. So this is used before DNA fingerprinting happens. So before the electrophoresis process happens, restriction enzymes are used to cut DNA into different pieces, different sizes, and it's always going to be cut at specific spots. So one example of a restriction enzyme is ECOR1, and this cuts DNA at wherever it reads AATT. That restriction enzyme will cut the DNA, just like you see in this image right here. Sometimes it makes a little zigzaggy cut like this one in this image, that creates sticky ends or just bases that are overhanging and don't have a partner anymore. Or sometimes the restriction enzyme will cut straight across, and that's called a blunt cut. It's just straight across that DNA molecule. So to practice this quickly here, the restriction site of ECOR1 again, or ECOR-I, is AATT. How many fragments will the following DNA strand be cut into? Okay, so every place where this restriction enzyme reads AATT, it's going to make a cut. So if it makes 2 cuts here, then 3 DNA fragments are going to be created. And then that can be placed, so these samples could then be placed in a gel, and electrophoresis can be done and separate these pieces inside of this gel. So this is what the setup for electrophoresis looks like. Here's the electric chamber. It has the gel inside of it, and you put the different DNA samples into those little holes that are at the top of the gel. If you look closely at the right-hand side of that gel in the middle of the box, you can see very dark blue DNA samples that have been stained, but that's where the little holes are located. Let's see, does this work? We don't really need the sound. I just wanted to show you it getting loaded. There we go. So here you can see the DNA getting put into these tiny little holes here in this gel. And then once those are all loaded up, then the electricity is applied. Come on, speed up. Okay, so electricity is turned on in this electrophoresis chamber. And then I just wanted to show you the end result. Bubbles. All right, there we go. So I think they have this set up as a little bit of a time lapse here. So then once the electricity is applied, the DNA moves down through the gel and it gets separated based on size. So the smaller pieces will be able to go farther in the gel, and then the bigger pieces will stay at the top of the gel. And these fragments, or these samples, must all be the same because they are moving at the exact same speed and distance. Ooh. Did not pause it quick enough. Okay, that's still very blurry, isn't it? There we go. So you can see here that these were all the same samples because they all ran the same distance here. But the pieces that moved the farthest to the right would have been smaller because they went farther in the gel. The bigger pieces would stay up closer to the holes, which is where they started. All right, so that's electrophoresis, and then what gets created is the DNA fingerprint, which you can use to compare different DNA samples. All right, we're almost done here. So last couple things here. So cloning is another part of biotechnology. If you're cloning an entire organism, or you could just be cloning parts of an organism, this is cloning. So gene cloning, that's in the middle there. It says making identical copies of a desired gene. If you're just copying one little gene, that's still cloning, but it's gene cloning. And then organism cloning, that's when you create an entirely new organism that is genetically identical to another one. So in order to do this organism cloning, here's an example. This diagram shows you, you can take the skin cell from one cat and the egg cell from another cat. With the egg cell, you take the DNA out of that one and take the DNA from cat number one, the cat you're trying to clone, put that DNA inside of the empty egg cell. And then you can put that into another cat to grow. Okay, so you're taking that DNA from cat one, putting it into the belly of the surrogate cat, and then she will then give birth to the cloned kitten. And so the cloned kitten, his DNA is going to be exactly the same as cat number one. So cat number one and then the cloned kitten, those are the clones. They have the same exact DNA. So there are pros to this. It can help create livestock with desired traits. It could maybe help replenish some of the endangered species that we have. Or if we're just cloning small genes that are producing useful proteins, we could then maybe use those to create some medicines that could be very beneficial for us. Therapeutic cloning, this is when we use stem cells to cure diseases or injuries. So stem cells are cells that are undifferentiated. They don't have a job yet, and they can be turned into any sort of cell. So if you look at this diagram here, for example, you can apply certain growth factors to stem cells to allow them to convert into blood cells or nerve cells, whatever is needed to do this therapeutic cloning process. Recombinant DNA, this is when pieces of DNA from two different sources are combined in one organism. The process of doing this is genetic engineering. And one example of this would be glowfish. So these glow in the dark fish have genes from a jellyfish put inside of them. And so now these once ordinary fish that did not glow now have a glow in the dark gene from a jellyfish inside of them, and now they glow in the dark. All right, last slide here. GMOs, you've probably heard of these, genetically modified organisms. These are any organisms that have recombinant DNA in them. So for example, those glowfish, those are GMOs. Those are organisms that have been changed genetically. And then the last thing, gene therapy. This is when recombinant DNA is used to cure human diseases. So sometimes people have different disorders as a result of having genes that aren't working properly. So the thought here is that by inserting a functioning gene into these cells, then hopefully the issue can be corrected. And with the good copy of the gene, then whatever protein was missing before can now be there to hopefully correct whatever issue was occurring for this person. All right, that was the last slide in our biotechnology notes. If you have any questions, make sure you are asking.